In today's video, I'll be explaining the methionine pathway with a focus of S-Adenosyl-Homocysteine, SAH, a byproduct of SAM, S-Adenosyl-methionine. I'll also include the roles of essential B vitamins and other crucial nutrients and how transfusion pathway fits in and affects the phase of homocysteine. Think of the methionine pathway as a sophisticated, highly regulated recycling plant inside a cell. Its primary purpose is to manage SAM, the universal metal donor which is essential for countless cellular processes, as seen over here. A metal group is a fundamental tag that can dramatically change a molecule's function. The enzymes that perform this transfer are called metal transferases. The human genome is predicted to encode more than 200 different metal transferases enzymes. It is not exaggerated to say that SAM is one of the most important cofactors in nature, second to ATP. SAM is required roughly 1 in 12 to 14 enzymes. Therefore, SAM has a very fundamental role in the chemistry of life. To keep this cycle active spinning, the methionine pathway has the options to either keep recycling homocysteine back to methionine or proceed into another pathway for the productions of our antioxidants glutathione, which is also another way for eliminations of clearance. Where it heads depends on various factors. The three major areas of metal transferase enzymes are productions, metabolism, and regulations. I will go over those enzymes which are very crucial with our day-to-day activities. So under the productions, we have this PEMT enzyme. PEMT catalyzes the synthesis of phosphatidylcholine, which is a very critical component for all our cell membranes inside our body. That's more than 37 trillion cells. Phosphatidylcholine is also needed for our bowel productions, and the optimal ratio is 1 part cholesterol to 10 parts phosphatidylcholine. Phosphatidylcholine also helps to package and transport triglycerides out of our liver via our VLDL cholesterols. The demand will also increase when making new cells, especially during pregnancy or if you are having sunburn. Then the second will be GAMT. GAMT catalyzes the final step in the synthesis of creatine. Creatine is very important for neurological support, muscle functions, and energy, and even speech. It affects our muscles and our body mass. It is also commonly occur as low birth weight for newborns. Low creatine levels slows down recovery, lower exercise tolerance, and gets fatigued more easily. It can also cause the body aches and pains. And since creatine plays a very critical role in brain energy metabolism, it certainly is a contributing factor to brain fog and cognitive performance. Speech delay development is also a very big thing. Think of those autistic kids when they are very young. They don't speak until on a later stage. I've included a link in my description box below for further reading at your own time. Then we have this PNMT. PNMT is a conversion of our stress hormones, which methylates norepinephrine to epinephrine. And then we have this ASMT, which helps to convert serotonin to melatonin, your sleep regulating hormones. So when you have low SAM, you might be having low melatonin, which will affect your sleeping quality. Under the metabolism, we have this HNMT, and this is the primary enzymes for inactivating histamine inside the cells in the central nervous systems and other tissues. Some examples of systemic histamines are skin allergies like eczema, running nose, and headaches. And there are also more serious cases like low blood pressure, irregular heartbeats, and even panic attack. If this histamine hits really hard on your central nervous system. Then you have COMT, which helps to metabolize catecholamines. This includes neurotransmitters and hormones like dopamine, norepinephrine, epinephrine, and estrogen. So when your COMT has a reduced ability to help to metabolize all these brain chemicals, then you will show high levels inside our body. So under regulations, we have GNMT. This is to convert glycine to sarcosine. These enzymes helps to regulate the SAM-SAH ratio, so to prevent excess buildup of the SAM or to act as a buffer. Then we have DNMT, DNA methylations. This is the primary mechanism for epigenetic control, turning genes off in a stable long-term manner, meaning that you switch off certain genes and maintaining genome stability. So let's go over a quick summary. While dozens of methyltransferase enzymes exist, the bulk of the body's SAMs is consumed by just a few high-flux enzymes. Approximately 80% of the SAM is primarily consumed by GMT for creatine synthesis and PMT phosphatidylcholine synthesis, with GNMT playing as a key regulatory role. And enzymes like COMT, PMT, and ASMT are pretty important for how our body works, but they don't use up tons of SAM overall. While methylation is the dominant function, SAM is also a precursor in a few other critical pathways, specifically these amino populations, the synthesis of polyamines, spermidine and spearmine, and the transferation pathway for the production of major antioxidants, glutathione, which I will discuss shortly. So let me take you step by step using the strategene pathway. Methionine is converted to SAM by MAT enzyme with essential cofactors indicated here. That's magnesium, potassium, and ATP. We all know that SAM serves as a primary metal donor in many methylation reactions as discussed earlier. Once the metal group is donated, it becomes SAH, S-Adenosyl-L-methionine. Since SAH has a chemical structure almost identical to SAM, it also a potent feedback inhibitor to metal transferases. So high level of SAH can inhibit metal transferase functions because it binds tightly to the active site of these enzymes competing with SAM. A high level of SAH means methylation is producing a lot of spent currency. The cell interprets this as a signal to pause methylations until this SAH is clear. SAH is then hydrolyzed to homocysteine and adenosine by this enzyme encoded by AHCY gene. This reaction is reversible, but the cell typically removes homocysteine to drive these reactions forward. And if you see the arrows here, when there's high homocysteine, it can actually combine with adenosine back to SAH again. Do also note that insufficiency of these cofactors like zinc and B6 over here can also raise homocysteine and cause this jamming of this junction. So let's go over the phase of homocysteine. Basically, homocysteine has two main phases. It can either be recycled or eliminated. Again, how much each of these phases are proceeded largely depends on many factors, such as the substrates available, the amount of cofactors you have in your body, the enzyme speeds, and all these inhibitors that can potentially slow down the enzymes. The remethylation back to methionine can occur via two pathways. And the methionine synthase pathway, which is the preferred route, is the primary pathway in most cells, which uses methylcobalamin, the active form of vitamin B12, as a cofactor, and the 5-methyl-tetrahydrofolate as the meta donor. So now let's put methylfolate and methylcobalamin into the picture. Methylfolate is the active form of folate, which we want at least 80% in our bloodstream. Methylfolate donates a metal group to homocysteine in the remethylation pathway via these enzymes, MTR, and these reactions require methylcobalamin as a cofactor. To make it clearer, there's a couple of reactions going on these junctions. First, methylcobalamin donates a metal group to homocysteine and turns into a reduced form of B12. Homocysteine will accept this metal group from methylcobalamin and becomes methionine, also known as methylhomocysteine. Then 5-methylfolate comes in and donates a metal group to the reduced B12 and becomes tetrahydrofolate. The reduced form of B12 will accept a metal group from methylfolate and becomes methylcobalamin again, so that it can actually join into the cycle again. Without adequate B12, these reactions will be impaired, leading to a buildup of homocysteine and a decreased activity in methionine synthase. It can cause what we call methylfolate trapping, where methylfolate is being stuck along here and gives an inconclusive B9 levels in lab reports. It also happened vice versa when methylfolate is deficiency, causing a normal or high level of B12. Both scenarios can potentially cause homocysteine level to rise and the methionine SAM levels to fall, crippling the entire methylation cycle. The second route for remethylation is via the BHMT pathway. This pathway as an emergency route. It uses betaine as the meta donor. Once donated, it becomes DMG. I bet you have also seen some individuals that uses TMG, trimethylglycines, a type of supplements, to help facilitate the job. But this minor route only happens mainly in the livers and kidneys. In the clearance phase of homocysteine, we have the pathway heading into the transfuration. Homocysteine can be converted to glutathione. This pathway involves two enzymes as shown here, CBS and CGL. And you also need cofactors like vitamin B6. This pathway is irreversible and disposes excess homocysteine. When more glutathione is needed to remove other toxic compounds such as your hydrogen peroxides, herbicides, and lipid peroxides, this pathway can speed up on its own. Besides processing toxic compounds, glutathione also plays a role in neutralizing hydroxyl radicals and nitric oxides, so that it can protect cellular proteins from irreversible oxidative damage. The balance between SAM and SAH is very critical. The SAM-SAH ratio is often used as an indicator of the methylation potential of the cell. The CBS enzyme is the first rate-limiting enzyme of the transfuration pathway. Its main job is to eliminate homocysteine into the productions of cysteine, glutathione, and other sulfur compounds. And the most important regulator of CBS is actually SAM. This is because SAM acts as an allosteric activator of CBS, which means that when SAM level is high, it signals that the cell's methylation status is good and there's an excess of methionine, so the body can afford to divert homocysteine away from the remethylations and into the productions of our antioxidants. But when SAM level is low, it signals that the methionine is much needed for methylation reactions. And the primary goal during low SAM conditions is to conserve methionine and replenish the SAM pool to support the vital methylation reactions. There are also other clinical and physiological implications that we need to put into considerations. The first one will be the insufficiency of nutrients. The proper functioning of these switches require adequate B vitamins. B6 is a crucial cofactor for CVS, whereas vitamin B12 and active B9 are essential for the remethylation pathways. We also need zinc over here. So a deficiency in any of these can disrupt this delicate balance. And these tags over here in purple, these are the inhibitors which plays a part in the kinetic space of the enzyme. Someone might suffer this hyperhomocysteinemia, which is a common genetic mutation of CBS, whereby the homocysteines fail to enter the transfuration pathway effectively, leading to a massive elevated homocysteine levels in the blood and urine, which can lead to serious clinical consequences. The supplement SAM is often used to support methylations and glutathione productions. Although this may sound doable for this mechanism, but the availability of these cofactors and other interplays between pathways have to be viewed as a whole picture. This also explains why some people don't feel the effect or even side effects after taking SAM. Do you see where it goes? Let's look at some of the symptoms associated with SAH feedback inhibitions. Each of these symptoms listed here are the cascaded effects of a poor methylation. Therefore, treating a symptom with a pill or supplement is not going to work. So if you see the first one, we have this high blood pressure, and it's usually linked with acid reflux, cold hands and feet, erythema. So if someone having this, you will go into the pathway and you will link to this bioptrin pathway, which will affect the productions of your nitric oxide. So when you have low nitric oxide, then you have all these symptoms. But if you visit a hospital or you go to your conventional doctor, your primary care provider, they will just treat each one of these symptoms as a standalone. If you have high blood pressure, they will have you taking medications to control your blood pressure. If you have acid reflux, the doctor might prescribe you with PPI. When you have cold hands and feet, they might say you have poor blood circulation, they may advise you to go for more exercise. And if you take a look at the second symptoms, we have insomnia, reduced sleep. As I mentioned just now, your sleep is largely affected by the amount of melatonin you have. Although you might be affected by how much stress you have, but they are actually all interlinked together. Then we also have histamine issues whereby you have a lot of skin related issues. You have eczema, psoriasis, dermatitis, you have your dandruff, all this. It's all related, closely related to your histamine pathway. How well your body can actually eliminate the excess histamine or the release of histamine. It's not about just eating something high in histamine food. Then you have this release of histamine. Yes, you may actually reduce the histamine bucket load by not eating this stuff. But the issue of having high histamine, that's another area which you need to look at. Then we also have a lot of ladies going for these anti-aging, wrinkles, fine lines, losing, or even to an extent of losing more hairs or gray hairs. These are closely related to the DNA productions. How well your body is managing your DNA, your cells, your skin cells. And outside the skin cells, you have membranes, and the membranes need your phosphatidylcholine. So if you go upstreams when you have your SAM levels low, you can't actually do the metal transferases enzymes to produce your phosphatidylcholine. And we also learned that phosphatidylcholine is also used in the productions of your bowel. So when your bowel flow is reduced, you have a lot of toxin buildup. It will cause you have poor detoxifications and poor fat absorptions. Then slowly over time you'll progress into some form of liver dysfunction. Then you have this mood disorder. You have anxiety, depressions, OCD, ruminations. All this is closely related to how well your brain chemicals is being produced, transported, and recycled and metabolized. Then we have this mitochondrial dysfunction. Imagine your body is a furnace which is burning the food you eat, the water you drink, the air you breathe. These are the inputs which will indicate how efficient your body is burning and produce the energy. So if you are not burning efficiently, or if you are burning with a lot of smoke, the smoke is what we call the oxidative stress. You also have poor digestions, malnutrition, because you're not absorbing all these cofactors, all these essential nutrients that your body requires to function all this pathway to keep this cycle spinning and spinning. And they are also closely related to each other. Like if you want to link poor digestions, it can be because you have acid reflux. You are low in stomach acid and you can't absorb all the nutrients. You can't break the food and absorb the nutrients. Then there's a bunch of nutrients that is needed to support these pathways. So insufficiency of all these B vitamins, other nutrients can cause your pathway to run very slow. The first thing that you can do is actually reduce all these purple text, all these things inside the purple text. You have to eliminate or reduce all these epigenetics that will potentially dirty your genes function. These are the folic acids, heavy metals. If you are staying inside a mold environment, you have to get out of it. Keep yourself away from more infections, your alcohol. You have to check your bacteria inside your gut, whether they are producing a lot of LPS, acetaldehydes, or ureas, which can cause to have this gut dysbiosis. When your body doesn't have all these inhibitors, you will have lesser reactive oxygen species. If you're taking some meds and drugs, first you have to know what's the underlying issues. Start from there. Then slowly work with your primary care provider and reduce the medications you are taking now. And the most important, you have to reduce your stress. Find what is causing you to have all this stress. It can be the environment, the air you breathe, the water you drink, or even if you are sitting on a table, on a chair for more than 8 hours, you need to reduce all this stress from your body to create lesser inflammations. This way, your body can actually burn more efficiently and produce more energy with lesser oxidative stress. So let's go over the summary. When the cycle is being charged up, methionine will convert into SAM. After the methylation has occurred, donated the methyl group, SAM will be converted to SAH. When high level of SAH, it actually puts a brakes to all metal transferases. SAH can be broken down into homocysteine and adenosine. Then the homocysteine can have two different fates. The first one will be the remethylations back to methionine, driven by methylfolate and methylcobalamin. So a deficiency here can cause homocysteine to accumulate. Then the second option will be this BHMT pathway, which recycle homocysteines using betaine as a meta donor, but is limited to liver and kidneys only. Then the second fate will be under this clearance pathway, which is the transferation pathway to produce glutathione. This can be activated by high SAM levels, and this pathway is non-reversible. Therefore, reduced activity of CBS, it will cause the transferation pathway to be effectively slowed down or put on hold. All these pathways interplaying each another is an elegant system that ensures methylations, a process fundamental to life, is precisely tuned to the cell's moment-to-moment needs. With SAH, is acting as a central report card to maintain perfect balance. CBS is regulated by a feedback mechanism in which low concentration of SAM stimulates more on the remethylations of homocysteines, whereas high concentration of SAM favor more on the transferation pathway. Okay, we have come to the end of this video. If you have not watched the video on how methylation works, do remember to click here.